Hello, welcome to chapter 20, Biotechnology part 2. Okay, we left off at DNA fingerprinting. So this is something that we can use in forensic science. So, like I said earlier, right? Oh, did not expect to be this far back. Okay, right here. We can see different kind of molecule, right? Based on its size. So the smaller one will be on this side. Yeah, the small one will be on this side, the big one will be on this side. So with forensic science, we can actually, let me go to the slide, use something. This is old technology, older technology, very old, actually, very old. We don't use it anymore. It's called RFLP, okay, rift lips. Okay, so what we do is we use the restriction enzyme to cut the DNA into different fragments, and then because every person has, even though we all have the same genome, but different expression of DNA would actually have different fragment. By looking at different fragment on gel, we can actually look for either diseases or if we want to identify a person. So this is outdated because it requires a lot of blood. Okay, so for example, right here, let's say we require a quarter size sample of blood, which is a lot. Okay, so what we do is we move on to something else. Well, actually, let me show you a picture. I forgot I sent this picture here. So we use restricted enzyme to cut at a very specific site. Okay, and then let's say if we have a normal person who has the beta globin like this, it's supposed to cut twice. However, if we if we have a sickle cell patient, it's only gonna cut once because there's a mutation right here. So as we can see, there is going to be two different large band, okay, and then you have one large band, two small band. So a normal sickle cell should have three bands, 1, 2, 3. The sickle cell patient would have two bands like this. So by looking at the electrophoresis gel, we can see that okay, this person has some kind of mutation based on its band mutation right here. So we can see this, and then a more updated one is called STR analysis. STR analysis, it stands for short tandem repeats. So what happens is we have observed that the non-coding DNA has a region of sequences that has repeating. Okay, so it's usually around 2 to 5 bases. However, every single person has a different numbers of repeat at different loci, so different location. Okay, so that's what loci mean. Okay, so the reason why we use this, well, remember the other one used like so much blood. Well, this one actually only use about 20 cells. So in a sample of a skin swab, you can probably collect way more than 20 cells. So that's the reason why we use this one. And this one also has very specific a number for person, depending on which chromosome you're testing. So this is a person's STR loci. So there's many different flag that we can double check and make sure everything is going well. So in forensic science, this allows us to see different peaks. So for example, these are different peaks of repeatings. And in forensic science, we actually saw that, well, this is a case, old case, that was recently, recently 2001, basically using the new technology forensic science, and then determined that, well, we actually have the wrong person. We did not have the right person. So by looking at the STR marker, so the semen on the victim, we see that, well, these two persons don't have the same marker, and marker 2 don't have the same marker. Marker 3 don't have the same thing, even though 12 match, but the 11 doesn't match. Well, the other person that they didn't catch actually has identical marker for all 3 locations. So that's how we determine that, okay, that would probably be the person who did the crime. So that's how we use biotechnology in the forensic realm. So these are some of the technology that I talked about today. There are a lot more detail that you need to read from the book. So, well, read the book. If you have any question, please let me know. This is chapter 20, Biotechnology, and this is part 2. And that's it for the lecture.